PGW students, I'm going to finish chapter 5, Inferring Phylogeny. And what we will do in class on Thursday is I'll do an example kind of towards the end, and we'll get what we think is the best phylogenetic tree using parsimony, and then I'm going to put up an alternative tree and then ask you to map what are called the character state changes onto that tree, and we'll see if it's more parsimonious or less parsimonious. And I think, given that I'm lecturing now, I'm going to hold off taking your homework, and I think we'll turn that in afterwards, or I might just post the answer so you can see it. So maybe I'll have time even in class on Thursday to go through one of the trees on the homework. Okay, so what we're going to do is talk about inferring phylogeny. And so we've looked at a lot of phylogenetic trees, but actually how do we reconstruct them is what we're going to spend our time, and how we use it to interpret. Now, phylogenetic trees are extremely important now and can be even used in forensic investigations. And this is an example of an incident in Benghazi, Libya. I think it was in the 2000s, maybe 1990s. I can't remember exactly. But there was an outbreak of HIV in a hospital in Benghazi, Libya. And it turned out that the initial charges were against 6 healthcare workers in the hospital, and they were charged with purposely putting HIV into the blood system or giving transfusions to patients, and that it was actually a plot by the CIA or the Israeli security forces to foment sort of unrest in Libya. And these people actually got convicted. They were supposed to be hung. And anyway, what people did is they actually developed a phylogenetic tree of HIV sequences, and that's what this is. And you don't see all the details, but you can see all these branch lengths. They're various sizes. And HIV evolves at an extremely high rate, and you can actually resolve relationships among all these different sequences and come to some conclusion. So here's what they found, and we're just going to be focusing kind of on the middle of this tree. There was a lot of evidence that these healthcare workers weren't actually involved in it, because it seemed like the infection in the hospital predated the arrival of these 6 healthcare workers. So there was a lot of suspicion, but that didn't seem to influence the decision to basically convict them and lead to their death. But so here's what they eventually found, was that it turned out that these were the sequences from about 44 patients in the Benghazi hospital, and they all cluster out pretty well. It turns out there's a fair amount of divergence between you don't see all 44 lines. Some of them have the same sequence, some are different, but they seem to have a fair amount of sequence divergence even among them, which suggests that they probably didn't occur. The introduction didn't occur from a single infection, or people doing these infections by infecting the blood supply or giving them transfusions. So there was some data there. The other thing that's more critical is that you can see that this Benghazi clusters actually with a group of HIV sequences that are common in Western Africa and the areas around Libya, Ghana, and Cameroon. And it turns out that the source of this particular infection, and this is what was demonstrated finally in court, and it took a lot of international pressure to show this, was that in fact what these HIV isolates probably were sourced from Ghana, and it probably came in through blood transfusion, and it was not purposeful. And so there was a lot of evidence, and there was no indication that the HIV was deliberately introduced by these six healthcare workers, and fortunately they were spared. So what I want to show is you can use phylogenetic trees to address particular hypotheses, and in this particular case it was used to exonerate these 6 healthcare workers that were accused of infecting patients in the Benghazi hospital. So what we're going to do today is we're going to kind of look at the general procedure for creating a phylogenetic tree, and there are a lot of different methods, and ranging from distance to what's called parsimony to what is called maximum likelihood. We're not going to get into all the details of the various methods by which we can construct phylogenetic trees, but we're going to primarily focus on how we use parsimony to reconstruct phylogenetic trees, and then how do we develop tests of particular hypotheses using trees, and how do we assess confidence in these tests. Okay, so here's an example of sort of 2 hypotheses about the phylogenetic relationships among, let's say, 4 populations or 4 different species. Hypothesis 1 is that species 1 and 2 are sister taxa and share a common ancestor, and 3 and 4 are sister taxa, and here's the root of the tree. Another hypothesis, and you can, out of 4 taxa, you can get multiple arrangements of these species in any sort of alternative phylogenetic tree, but another alternative phylogenetic tree is that 2 and 3 are sister taxa at the tips of the branch, and then they are more closely related to 4, and 2, 3, and 4 share a common ancestor, and 1 falls in the outside. Okay, so these are various hypotheses for how these species could be phylogenetically related to one another, and we can actually do a lot of things to look at various kinds of character states and what is the most simplest, most parsimonious explanation for the relationships. And parsimony is a concept, kind of a philosophical concept, that suggests that the simplest, least number of steps is the solution or the phylogeny that we tentatively accept. And of course, new data can always change that, but under the current data set, this is what we believe. So let's imagine we have these various hypotheses, and these hypothesis 1 that we showed before, what we're saying is species 2 and 3, in both of these hypotheses, both have a dark coat, and 1 and 4 have a yellow coat. So there are 2 ways that you can get sort of the evolution of either a dark coat or a light coat, given that hypothesis 1, where 1 and 2 are sister taxa, 3 and 4 are sister taxa. So dark coats, what you could have is the ancestral state could be light coat, and along this branch it changed to a dark coat leading to 2, and it independently changed to a dark coat. So there's another independent origin of the dark coat on this branch leading to 3. So this required 2 evolutionary changes to dark coat, and they are independent of one another. So the dark coat evolved independently twice. Okay, and you can kind of think about the mouse example or a lot of different examples of convergent evolution. The other possibility, of course, is the ancestral state was a dark coat, and in species 1, it changed to a yellow coat independently, and it changed to a light coat in species 4. Irregardless of what was the ancestral state, and it could be either way, and we'd need what's called an outgroup to determine this, and we'll come to that, what the ancestral state was. This particular tree, where 1 and 2 are sister taxa and 3 or 4, for this particular trait requires 2 ticks or 2 changes on the tree to account for the evolution of the coat coloration. Okay, let's look at hypothesis 2. Hypothesis 2 is the case where you would say that yellow is the ancestral character state, and then the dark coat arose in the common ancestor to 2 and 3, and it only had to arise once. So all you need is the dark coat to arise in the common ancestor 2 and 3 to get the coat color to map correctly onto this tree. So this is a case where only a single evolutionary event or origin of dark coat was required rather than 2. So we would say this is the more parsimonious explanation, and for this particular character, this phylogenetic tree is simpler and more parsimonious and fits the data better. Okay, let's go into a little more detail about how we do these sorts of things, and we'll be doing examples of this, but what we have here, this is a little bigger tree. Now we have 5 species, 5 branches, and on the terminal branches, we have a bunch of different characters, and let's just say this is some sort of coloration or whatever you want to call it. This is dark purple, and these are light purple. So species 1 is dark purple. Species 2, 3, 4, and 5 are all light purple. This is one character. This is the second character. Here's the third character. For the greenish character, 3 of these species, 1, 2, and 3, have a dark green, and 4 and 5 have light green. On the other hand, here, 1, 2, and 3 have light blue, and 4 and 5 have dark blue. Okay, so there are lots of ways to do this, and what we're going to do is we're going to kind of map these character state changes onto particular trees, and what we're going to do is we're going to be counting the number of evolutionary changes on the tree and determining how many evolutionary changes have occurred and how many character changes have occurred. Okay, so let's start down here. Oftentimes what we do in phylogenetic trees at the base, we have to infer the ancestral state. This is, for this example, this might be what we would call the outgroup species. And these 2, 3, and 4s, we're trying to determine the relationships in the in group. So let's just say this is the out group, and we've inferred the inferred ancestral state for all 3 characters is light blue, dark green, whoops, and dark purple. Okay, so what we're going to do for this arrangement, where 3 and 4 are sister taxa, 3, 4, and 5 share a common ancestor, and then 2, 3, and 4 share a common ancestor, we're going to map the changes in these character states along this tree. So let's go right here. How did we get this? We put a tick mark here where P, which was big P was the ancestral state, changed to small or light purple. Okay? And the reason we do that, we put it on that branch, is you go up here and look at this character. All 4 species, 2, 3, 4, and 5, have the light purple or small p. So all that's required is that it evolve from dark purple or big P to small p, and all 4 species have retained that small p. So we only need that tick mark there. Okay, let's look at another character that might require a couple changes. Okay, let's look at small b. So what's happened here? So what's happening here is, what we're saying is, what happened here is that we mark this here where small b changes to big B. Okay? And the reason we do that is it goes up here and you can see this has big B here. Okay? We go up here and this is still big B. Okay? So by putting that tick mark there, you can account for the presence of the big B changing, the change from the ancestral trait of small b to big B along this branch, and these 2 species have it. Now, to account for this though, right, you have to have this reversal back to the ancestral state. And this is a case where what happened is it changed to big B here, everything's fine, and then to get the small b in 3, you have to change it from a big B to a small b. So that's called a reversion to the ancestral state. So that particular character required 2 evolutionary changes to account for this. Okay. Again, let's look at green. So in this particular case, and there are a couple ways you can do it, this involves saying that these both evolved independently, where the ancestral state was G, and along this branch, remember this species has a small g, so we put a tick mark there. It changed from G to small g along that branch. And the other possibility, this is a small g, so it changed from big G to small g on that, and this has the ancestral state in it. Okay, so there are other ways to do this, and this is my preference, is that you always sort of independently mark the origin of a new character state or a derived character state. Okay, so the key point here is for these 3 characters and these 2 different states of each character, this particular phylogenetic tree has 1 mark, 2 marks, 3 marks, 4 marks, and 5 marks. The number of steps in this particular tree are 5 evolutionary changes. So the question is, this is just one of many possible trees for the relationships among 2, 3, 4, and 5, with 1 as the outgroup. There are other possible trees that you can get from this, and let's just look at maybe another arrangement what we get. Okay, so here, instead of this case where 3 and 4 were sister taxa and they were more closely related to 5, here we have another tree where 2 and 3 are sister taxa and 4 and 5. So this is just an alternative phylogenetic tree, and let's see how many evolutionary changes are required for this particular tree. So again, this is all the same. Big P is the ancestral state. It changes to small p in all 4 species, 2, 3, 4, and 5. Okay? Now, what we can see here is the ancestral state for the B character is a small b, and you put this tick mark along here because only 4 and 5 have the derived character state, big B. These 2 have small b. So all you need is one evolutionary change from small b to big B on this branch, and the same is true for the ancestral character state of big G to small g, only has to change here, and so that can account for the 2 small g's, and these have retained the ancestral states. Okay, so what this says is for this particular phylogenetic tree, you require 3 evolutionary steps, and this is what we would call a simpler, more parsimonious tree than that one, which has 5. And what we would suggest is this is a better phylogenetic tree than the previous one. And in fact, 4 and 5 share 2 derived character states in common with one another, are sister taxa, and 2 and 3 are sister taxa, although there's not much support for that. Okay, so that introduces kind of the concept of parsimony. Okay, we can also, and this is a fairly complicated graph and we'll go through all of it. It's real easy using DNA sequence data. And again, we're given a phylogenetic tree here where C and D are sister taxa, E and F are sister taxa, G and H are sister taxa, and A and B. And E, F, G, and H share a common ancestor, and these all. So these are kind of the outgroups. Okay, so you'll be doing some of this for your homework, and we'll be doing some examples in lab. So I just kind of want to introduce how you mark changes in nucleotide sequence and for different kinds of characters. So here you have a whole string of DNA sequences for species A, B, C, D, E, F, G, and H, okay? And here are the various nucleotide sites. So what we're looking at primarily are the ones that are highlighted, are the ones that are variable. You can see this is not a variable site, this is not a variable site, it's not informative, it doesn't help us, none of these. So we're primarily looking at the color-coded ones, and this is where we've seen some evolutionary changes in the DNA sequences of these species. So let's look at nucleotide site 1. What you can see is all species have C, except C and D have a G. So obviously you would change, presumably C was the ancestral state, change to a G in species C and D. Here's another example for the blue coloration. The character number 2, A and B, have C, and all the others have A. So if you go down here, here's where we can put this. We can have A change to C in the branch leading to species A and B. A and B both have C in common, and all the others have A. And not to belabor the point, but here we'll do another one. Here's 3, where all 4 of these species have C. This one has T, and presumably this might be the derived state. So where would E, F, G, and H? So here's E, F, G, and H. You can go back to here, and in this one you can see that it changes from C to T, the ancestral state, from C to the derived state T, in common ancestor E, F, G, and H. So you can do this for all these characters, and you might want to just do this as an exercise, and you can map these character state changes on this tree. And again, this is probably the best one, but we could arrange all these taxa in a lot of different ways where C might be related to B, A might be related to G, and we can do a bunch of alternative trees. But for this particular tree, for these 7 variable nucleotide sites, here's what we have. These are the character state changes at each one of these nucleotide sites. And in this particular tree, there would be 1, 2, 3, 4, 5, 6, 7 nucleotide changes. And it's probably the best tree. But again, we'd want to develop some other method for doing that. Okay, here's an example of how we can use phylogenetic trees to understand the evolutionary history and diversification of particular kinds of species. And magpies are kind of related to crows. I think they're in the Corvidae. And they're very common throughout Asia, Europe, and these are the Korean magpies and the Eurasian magpies. Are believed to be fairly closely related. And then the black-billed magpie and the yellow-billed magpie are North American magpies and have probably recently colonized North America from Eurasian ancestors. But how do we address that? So one of the things we don't really know as we look back at this tree, who's more closely related to whom. So, well, it looks like they do share a common ancestor, Korean magpie and Eurasian magpies and black-billed magpies. But if you put the root here, it might be a little different. So one of the things we want to be able to do is determine kind of the root of the tree. And one of the things that's often done is, as we've talked about before, if this is kind of the phylogenetic relationships and we want to convert it into this, we use an outgroup species. And so this could be the azure-winged magpie is a related magpie, and it's being used here as the outgroup to all 4 of these species. And it's basically used to kind of develop what is the root, and this is the outgroup, and then determine the relationships among the 4. So when you root it here and you put that branch there, it then converts into a tree in which the Korean magpie diverged first, and then the Eurasian magpie diverged from that common ancestor, and then the black-billed magpie and the yellow-billed magpie share a common ancestor, and the divergence occurred here. So a lot of times what we can do is this, we kind of infer the speciation process by looking at these phylogenetic trees, and we can use a lot of other information to address this. Okay, so one of the things you may have noticed is of all the characters that you see in all these species, from the azure-winged Korean, Eurasian black-billed magpie, the one character that's unusual is the yellow bill, and seems to be the yellow bill only occurs, obviously they named it that, the yellow-billed magpie. So how do we, given this phylogenetic tree, explain the evolution, and how often did those yellow beaks originate? Okay, so this seems kind of obvious, but it's in your book, and it's the other thing. So one of the things that we could do is we could say that the ancestral state, one hypothesis is the ancestral state of the bill is yellow coloration. If you do that, given this particular tree, black beaks have to originate independently 3 times along each one of these branches if the ancestral state was yellow. So if the ancestral state is yellow, then you have to have the evolution of black bills occur 3 times independently along each one of these branches. A more parsimonious explanation, obviously, is that the ancestral state is black. Okay, I don't know why this is green, it should be black, but the ancestral state is black, and it really only, yellow beaks only originated once if the ancestral state was black in the species giving rise to the yellow-billed magpie. Okay, the other thing you can do by studying this phylogenetic tree and looking at the geographic distribution of these magpies, and there's a lot of other evidence for this, primarily from the azure-winged magpie and where other magpies are present, is that we are pretty sure that magpies originated somewhere in East Asia, and that's believed to be. And here are the geographic distributions of all the various species. So in green is the Eurasian magpie, in blue is the Korean magpie, in purple is the black-billed magpie, and in yellow is the yellow-billed magpie. So given this phylogenetic tree, that what we can basically hypothesize is magpies originate somewhere in East Asia. Then you follow this up, there was a divergence or split or a speciation process separating the Korean magpies and the Eurasian magpies, okay? And this is believed that basically Korean and Asian magpies split off first, okay? So how did we then get the black-billed magpies in North America? And I don't know the exact dating on this, but it's probably occurred sometime in sort of between Pleistocene glaciation events, and North America wasn't very hospitable at certain times, but it's believed to be that a subpopulation of the European magpies, so the European magpies and the black-billed magpies and the yellow-billed magpies all have a common ancestor. So it's believed that a subpopulation of the European magpies, basically from probably this region over here, crossed the Bering Strait into North America, and then that led to North America then being split into yellow-billed and black-billed species. And the yellow-billed species is primarily in the western United States. The black-billed is here, and there's some degree of geographic isolation, and so it's believed to be that the yellow-billed magpie and the black-billed magpie split sometime during the Pleistocene. So again, all we really need is one change in the character state in terms of the coloration, but we can also understand how these species originated, that they originated somewhere in East Asia, they split into these 2 species, and then subsequently they moved into North America and split into 2 other species. So clearly each one of these things represents a speciation event. Okay, so now what I want to do is I'm going to go to the parsimony example and kind of go through how we actually do this from original data. And what I'm going to do is I'm going to stop the recording here and create another recording that is just the parsimony example, so you can go to that directly.